Welcome to Professore Attilio Scienza's newest book, translated and narrated by Richard Huff. This latest publication is part of the Mama Jumbo Shrimp series, entitled Mama Jumbo Shrimp Guide to Vine and Prejudice, Fake Science and the Search for the Perfect Grape. This is a great way to get a sneak peek at the book before it hits the shelves, so listen in and let the geeky knowledge seep into your ears, because we all need a little bit of Scienza.
Chapter 7. The Proto-Pinot, a glacial relict. The wild grapevines of the island of Ketsch in the German Upper Rhine represent one of the largest remaining populations of Vitis sylvestris in Western Europe. Scientific studies have shown these vines to be genetically close to the Basque wild vine populations and similar to those of Caucasia. The findings suggest that the Ketsch population may be a relict of wild grapevines that survived from the last ice age. They may, in fact, be considered to be native European wild grapevines. Pinot originated from these wild plants, the results of a spontaneous crossbreeding that took place about 2,000 years ago, when vines of Asiatic origin were brought by the Greeks from the colony of Massalia. It is possible to find traces of this introgression in their DNA. The presence of an allele that codes for the proteins responsible for the synthesis of anthranilite derivatives in the ancestral pinots of Ketsch Island is attributable to the genetic isolation of that small population that has survived since the glaciation. The promoter of the gene responsible for this trait was probably methylated by an epigenetic event that blocked its activation. Many genes are modulated in this way, and many corresponding traits depend on this process. This also explains why this strand of DNA was not passed on to the numerous Pinot offspring. Chapter 8. American and European Vines. A common history? In the scientific community, the common origin of the American and the European species of vine remains a divisive issue. Classifications based on ampeleographic and geographical characteristics clearly differentiate between the species, rather like comparisons made in the past between human races. The proof of these differences, according to the defenders of the purity of the European vine, is the presence of two metabolites in the berries: the ester of methylanthranilite, which is responsible for the strawberry flavor, and malvidine-3 glucoside, which is particularly present in red grapeskrin and red wine. But can two chemical compounds in berries really be responsible for the differences between the American and the European vine? As a result of a double mutation, the gene that codes for the malvidin is underexpressed in Vitis vinifera, although it is present in its genome, and is related to a gene involved in the synthesis of the characteristic foxy aroma of Vitis labrusca. This colocalization means that in the hybrids of Vitis labrusca, the foxy aroma is transferred together with those of synthesized anthocyanins. However, due to the effects of genetic mutations or recombinations that occurred during evolution, the expression of these genes has, over generations, been profoundly changed in the European vine, which is why the foxy aroma is no longer so apparent. This absence can be attributed to the genetic drift due to the reduced number of individuals in the population, or to the effect of the reduction of variability caused by endozoochory, the bottleneck, the biotic dispersal of seeds performed by birds who are attracted to plants with large, colourful berries with high contents of sugars and other aromatic components. Environmental conditions, for example, low temperatures during particular periods of the reproductive cycle, may have also induced the expression of some otherwise silent genes. This interaction with the outside world is explained by epigenetics, the process of gene modification that does not alter the gene sequence, but is instead modified by mutations caused by the adaptation mechanism of the plant in response to environmental stimuli such as high radiation, water and thermal shocks, food shortages, etc. In relation to the advancement of varieties with particular phenotypic characteristics and in their subsequent multiplication, humans can intervene through clonal selection, and birds, in the case of wild plants, through the choice of berry that they feed on and disperse. On this basis, it is possible to understand why the chemical compounds responsible for the foxy or strawberry aroma, methylantranilate, in the berries of European vines are present in much smaller quantities compared to the American vines derived from Vitis labrusca. The component of the gene that contains this strawberry aroma is present in American grape varieties because of a genetic mutation that occurred over time. In that context, the distinctive flavors probably encouraged the widespread dispersal of the seeds by certain animals who are attracted to the fruit by its pungent aroma. There may also be another factor linked to the role that these compounds may have in the defense of the plant from insects and/or fungi. The presence of methylantranilate in Vitis labrusca and Vitis rotundifolia and their relative absence in other grape species is a form of adaptation that has encouraged seed dispersal. These odorous compounds may in fact have been produced by the vine to protect the berries from attacks by birds, since the smell of the molecule, reminiscent of jasmine, may also act as a repellent. At the same time, they could also function as an attractant for other animals, including small rodents, due to their preference for seeds whose integuments are rich in tannins and need to be digested by gastric juices to germinate. On the other hand, in Europe, since this evolutionary mechanism was less prevalent, the gene frequency responsible for the synthesis of these substances was lower. One of the most important aspects of the specificity of American species is the synthesis of anthocyanin glycosides. This specificity is transmitted by a dominant strain of DNA, which, due to a double mutation, is no longer found in Vitis vinifera. The gene in question is located on chromosome 9 of the Pinot Noir genome and is related to the putative gene of an alcohol that is involved in the synthesis of the characteristic foxy aroma of Vitis labrusca. This co-localization means that in Vitis labrusca hybrids, the foxy aroma is transferred together with that of the synthesis of anthocyanin glycosides, but also that due to the effects of genetic mutations or recombinations, the expression of these compounds can appear profoundly changed over generations. In the makeup of the foxy aroma, there are at least three chemicals: methylantranilate, 2-AAAP, and furanianol, of which methylantranilate is the most important compound. Although associated with American species and their hybrids, MA has also been detected in Pinot Noir, along with esters of antranilic and cyanic acids, and is the basis of the typical fruity notes, exotic fruit, dried plums, strawberries, berries, of Burgundian Pinot Noir. The levels of methylantranilate detected in the grapes descended from Vitis labrusca can vary between 20 and 30 grams per litre. In red wines obtained from the vines of Vitis vinifera, the maximum detected is 100 micrograms per litre. Concentrations ranging from 4.1 to 823 micrograms per litre for furaniol and from 9.5 to 380 micrograms per litre for methylantranilate have been detected. The detection limits in hydroalcoholic solutions of furaniol and methylantranilate are 37 micrograms per litre and 3 micrograms per liter respectively. Furaniol is the most abundant aromatic compound in the sensory profiles of Vitis rotundifolia and an important component of the characteristic aroma of Vitis labrusca. However, furaniol has also been identified in numerous cultivars of Vitis vinifera, such as Tempranillo, Grenache, and especially Pinot Noir. The concentrations are much lower than the Vitis rotundifolia and Vitis labrusca hybrids, but can still significantly exceed the detection threshold of 5 to 37 micrograms per liter and contribute to the fruitiness of Vitis vinifera wines even at low concentrations. Furan derivatives more common in Vitis vinifera wines, such as furfural and sotolone, are formed during fermentation and aging. Methoantranilite forms the basis of the atypical aging off flavour in some wines obtained from Vitis vinifera. It is commonly recognized as giving the smell of wet wool, furniture polish, or mothballs.
Listen to the Italian Wine Podcast wherever you get your podcasts. We're on SoundCloud, Apple Podcasts, Spotify, Himalaya FM, and more. Don't forget to subscribe and rate the show. If you enjoy listening, please consider donating through ItalianWinePodcast.com. Any amount helps cover equipment, production, and publication costs. Until next time, Cin Cin.
Guys, I'm Joy Livingston, and I am the producer of the Italian Wine Podcast. Thank you for listening. We are the only wine podcast that has been doing a daily show since the pandemic began. This is a labor of love, and we are committed to bringing you free content every day. Of course, this takes time and effort, not to mention the cost of equipment, production, and editing. We would be grateful for your donations, suggestions, requests, and ideas. For more information on how to get in touch, go to ItalianWinePodcast.com.